Assalamu alaikum, I am Rufia Abbas on behalf of Sindh Biotechnologist Association. I am going to teach you genetic engineering in this lecture. In these slides, we will discuss genetic engineering, applications of genetic engineering, ethical issues related to genetic engineering. For further explanation, let's move to first slide. Genetic engineering. Genetic engineering is the artificial manipulation or alteration of genes. It develops in 1970s. Genetic engineering involves removing a gene from one organism, inserting target gene into DNA of another organism by cut and paste process. It also involves in artificial synthesis, modification, removal, addition and repair of genetic material. We can obtain millions of copies of species DNA inside a bacterial cell. Some important terms used in genetic engineering. Recombinant DNA. The altered DNA is called recombinant DNA. It involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine DNA from different species or to create genes with new function. The resulting copies is recombinant DNA. The next one is GMO. Genetic modified organism is an animal, plant or microbe whose DNA has been altered using genetic engineering technique. GMOs are used to produce many medications and genetically modified foods and are widely used in research and for production of other goods. Genetic engineering breaks the species barrier. It allows DNA from different species to be joined together. This often results in combination of DNA that would never be possible in nature. For this reason, genetic engineering is not a natural process. If DNA is transferred from one species to another, the organism that received the DNA is said to be transgenic. Genetic engineering can be done on any living organism because all living organisms contain DNA within each cell nucleus. Examples of cross-species transfer of genes. A human gene inserted into a bacterium, a human gene inserted into another animal, a bacterial gene placed in a plant. Applications of genetic engineering. We must know three applications, one involving a plant, one animal, and one for a microorganism. The first one is plant. Weed killer resistant crops. Since 1996, GM crops or GM plants have become an important component of our food supply. GM crops are transgenic plants that resist pests, herbicides, disease, and result in increased yields. Weeds are unwanted plants that grow along the crop plants. It is estimated that crop yields is reduced by 10 to 15% due to presence of weeds. To tackle down the problem of weeds, many types of crop plants have bacterial gene added to them. These genes make the plant resistant to certain weed killers. This means that the weed killer kills the weeds but do not affect the other transgenic plants. Genetic engineering in animals. Transgenic animals are becoming more important to our food supply. There is a growing trend to experiment with inserting human genes into the DNA of other mammals. The transgenic animals formed in this way will then produce the human proteins and secrete it into their milk or even into their eggs. Milk prevents infections in young children who drink it. Sheep produce human clotting factor. Genetic engineering has been applied in both sheep and goats for various purposes to investigate the biological and functional roles of genes, to introduce novel economical important traits for agriculture purposes to produce valuable proteins in milk. A human gene has been inserted into the DNA of sheep. This allows the adult sheep to produce a clotting factor or a clotting chemical needed by hemophilia to clot their blood produced in the milk of sheep. Genetic engineering in microorganisms. Bacteria make insulin. The first genetically engineered synthetic insulin was produced in 1978. The human insulin gene has been inserted into a bacterium E. coli. This allows the bacterium to produce insulin for use of diabetes. Ethical issues in genetic engineering. The first one is the release of GMOs into the environment. If GMOs are released into the environment, there is a possibility of danger. They can grow in bioreactors, do not provoke as much fear. If GMOs are grown outdoors, there is a fear of foreign genes they contain separate to the other plants. It had negative impact on traditional farming practices. The second one is animal welfare. The generation of new genetic engineering line of animals often involves in the sacrifice of some animals and surgical procedures, example surgical embryo transfer. There is a serious concern that animals will suffer as a result of being genetically modified. Use of growth hormone may cause limb of deformation and arthritis as animals grow. Genetic engineering in humans. In human, genetic engineering is used to treat genetic disorder and cancer. It also helps in supplying new body parts. Although this has not been done today, genetic engineering has potential of creating new types of human beings with many advantageous traits. Genetic engineering is used in field of mining to extract useful elements. Thank you so much for your attention.